And the first speaker, it's my great pleasure to introduce my Canadian colleague, Dr. Leonardo Cortez from the Center for Prions and Protein Misfolding Diseases at the University of Alberta in Canada. And Leo is going to be talking about the grant he received entitled Prion Protein Aggregate Size Distribution Drives Clinical Phenotype. Leo.
Okay, hi everybody. Thanks for having me, and thanks all for the introduction. So yes, today I will tell you about how we take advantage of prion size to better understand the clinical presentation in sporadic CJD. Oh. Okay, let me see how. Okay, yeah. So as you know, in prion diseases, we have accumulation of prion particles in the brain. And these prion particles have a wide variety of sizes and shapes. So usually when we study prions, we take a portion of the tissue that we are studying, and we analyze all the prions that are present in that tissue. So at the end, we have an average representation of what is happening in the brain. But if we want to have a better picture of what is happening in the brain, and that is important to understand better the disease, it would be ideal to separate different population of prions that share similar properties. And that's a complex task. But in our lab, we have a method that is called asymmetric field flow fractionation, or AF4, that allows us to separate different population of prions based on their size. So we can put in different tubes prions of different size and analyze them individually. And one method that is currently or normally used in the lab to analyze prions is Western blot. And Western blot will give us some information about the prions that we are analyzing. So it can give us information about the amount of prion that we have, and sometimes also the type of prion that we have in the tube. So Western blot is a kind of fingerprint of the prions that we are studying. We decided to apply, with the support of CJD Foundation, we decided to apply this approach to the study of sporadic CJD prions. So as you heard during the talks this morning, so sporadic CJD is the most common form of CJD. It represents almost 85% of total CJD cases. And sporadic CJD have a wide phenotypic variability regarding age of onset, rate of progression, neuropathological features, and also different depositions of prions in different parts of the brain. A lot of effort have been invested trying to understand this variability and also trying to classify the different subtypes of sporadic CJD. And the current sporadic CJD classification recognizes at least 6 clinical pathological phenotypes, 6 clinical presentations of CJD. And this classification is based in 2 facts. One is the polymorphism at codon 129, and the other fact is the type of prion that accumulates in the brain. So the polymorphism at polymorphism are normal variation that we have in our genes. And specifically in the gene for human prion protein, we have a polymorphism at position 229 that make our normal prion protein to have 2 different possible amino acids, methionine or M and valine or V. So we can have in our protein normally a methionine or valine at this position. But because we have 2 copies for this gene, we can have the 2 copies with methionine, 1 copy with methionine and 1 with valine, or 2 with valine. So those are the 3 possible combinations that we can have for this polymorphism. The other aspect for this classification is the type of prion that we have, or that the CJD patient have. So, and like I mentioned, so in the brain, different forms of prion are accumulated. Some of these forms are more compact, and some are less compact. If we treat these prions with proteinases, proteinases are molecules that degrade proteins. So the proteinases cut the proteins in small pieces. Well, if we treat these prions with proteinase, and one of the most common proteinases that we use in the lab is proteinase K or PK, we will degrade the prions that are less compact, and we will have at the end of this process only the most resistant prions. We call those resistant prions PK-resistant prions, and the prions that are degraded we call PK-sensitive prions. So now we can analyze by Western blot the PK-resistant prions, and we will have a fingerprint of that resistant core. So, for example, we can have a type of prion that we can call type 1 and run a Western blot and have their fingerprint with specific spots. One of those spots could be one band or one spot at 21 kilodalton. Kilodalton is the way that we measure size of proteins, right? But like I said, different phenotypes, different clinical presentation of the disease can accumulate different type of prions. So we can have type 1 and type 2. And if we analyze those type of prions by Western blot, we can have different fingerprint for those type of prions. So if we combine the type of prions that we can have in sporadic CJD with the polymorphism, we have these 6 different combinations. MM1, MB1, BB1, MM2, MB2, and BB2. But like I said, so we believe that to have different clinical presentation, we need a different type of prion. And here we have 6 different type of presentation of the disease with only 2 type of prions. Why that is happening? One reason that can explain this is that the technique that we use to classify the type of prions is not sensitive enough to distinguish different or small variation between all these types of prion that accumulate in the different presentation of the disease. So we wonder if using our asymmetric field flow fractionation, we can see in more detail the variation in the type of prion that are present in each of these subtypes. So for this presentation, I will only show you the comparison between MM1 and MB1. So if we do the traditional analysis, we will see in a Western blot that MM1 and MB1 have the same pattern in a Western blot with that spot at 21 kilodalton. But what happens if we take that sample, separate the prion by size, and analyze by Western blot? That's what we've done. And here is the Western blot for the different fractions that we collect. That means the different tubes with the different, with the prions of different size. And then we treat those samples with PK, and we analyze those PK-treated samples by Western blot. And what we can see here, if we compare side by side, is that now MM1 and MB1 have a different profile in a Western blot. So different fingerprint, which may be indicating that we actually have different type of prion in these 2 samples, right? So we compare also with MM2, type 2 of sporadic CJD, just to have a control for these samples. What I showed you before is samples from one area of the brain, that's frontal cortex. We did the same in other areas of the brain. Here is thalamus, and we observe exactly the same. So that MM1 and MV1 have different fingerprint, different profile in a Western blot. So what we think at the beginning that is the same type of prion, maybe is a type of prion. So MM1 and MB1 have subtle difference in the type of prion. So we wonder if we should call them, both of them, type 1. Other aspect that we analyzed is the total amount of prion present in those samples. So when we treat, like I said before, when we treat with PK, we are only analyzing the PK-resistant particles. what happens if we don't treat those samples with proteinase K. So we did the analysis without the PK treatment, and in this analysis we can see or compare the amount of proteinase-sensitive prion particles and proteinase-resistant prion particles. So in this Western blot, you can see that in the first lines from 4 to 19, we have the prion-sensitive, the PK-sensitive prion particles, and in the fraction with larger particles, we have the PK-resistant prions. So what we found when we compare MM1 and MV1 is that the proportion of these populations is different in MM1 and in MV1. We check that in different regions of the brain and also compare with MM2. So in conclusion, what we saw in this part of our project is that MM1 and MV1 could have different type of prions, and also the population, the proportion of the PK-resistant and PK-sensitive population is different in both cases. And that can explain the variability in clinical presentation of the different subtype of prion protein. So seeing those results, we decided to see what happened if we compare now in the same subtype of prion, in this case MM1, different regions of the brain. And that is important because if different regions of the brain are affected differently, we will have also different clinical presentation of sporadic CJD. So we analyze frontal cortex, thalamus, and cerebellum of MM1 sample. And here again, we observe some difference. So in the same subtype of sporadic CJD, we observe that frontal cortex and thalamus have the same type of prion, but cerebellum have a different profile in the Western blot, suggesting that there is other type of prion in cerebellum. We also analyze the population of PK-sensitive and PK-resistant prions, and we also see a difference between these two populations. Now we started comparing also the replication activity, so how well these prion particles replicate when they come from different areas of the brain. We're still trying to adapt the protocol to have better results, so far we don't have conclusive result for this part. And we are also analyzing the lipidic composition of the different parts of the brain that have different type of prions. So the prion conversion occurs in the cell membrane, and the cell membrane is composed of, is a very complex structure composed of different type of lipid. So we did the lipidomic analysis, so we analyzed how much of each different lipid is present, for example, in cerebellum, frontal cortex, and thalamus of patients with sporadic CJD, and try to correlate this difference in lipid composition with the different type of prion that we are seeing. So as a summary, we can say that sporadic CJD can present with variable phenotypes, and presumably these different phenotypes are related to different type of prion. But the current method to discriminate sporadic CJD neither capture nor explain the full diversity of these phenotypes. So with the aim of understanding the phenotypic diversity, we use asymmetric field flow fractionation to isolate and characterize prion particles accumulated in sporadic CJD brains affected by different types. And we have identified strain-specific prions particles from different brain regions. Our comprehensive analysis of all prion particles in sporadic CJD brains will contribute to better understand of the role in driving phenotypic diversity. So with all, that's all my presentation, and I want to thank all members of my lab, especially my supervisor, Valerie Sim, Satish Nemani, who have done most of, or lots of the Western blot presented here, also the CJD surveillance system, And special thanks for all families, and specifically for yes, the contribution of Jeffrey and Mary Smith family for supporting this project. So that's all.